Okay, I think we can get started.
Hello everyone, welcome to our webinar today. My name is Elaine Zhang, Technical Support at GemPharmatech. It's my great pleasure today to introduce my colleague Zhiying Li to share her experience and knowledge in germ-free mice for microbiome research and drug development. As a brief intro, Dr. Li is a neuroscientist by training. Her PhD study focused on synaptic transmission and pre-synaptic vascular recycling, which was conducted in the lab of Dr. Zactek N. Mercy at Harvard University. She did her postdoctoral training in Dr. Jeffrey Friedman's lab at Rockefeller University, where she studied cell type-specific hypothalamic networks of metabolic regulation in the context of leptin signaling. Afterwards, she joined Technical Bioscience, focused on genetically engineered rodent models for health and disease, supporting genetic science and compliance, as well as scientific and commercial production. She joined GemPharmatech US as Director of Life Science and Technology in October 2021. Without further ado, welcome Zhiying, and the stage is yours, Zhiying.
Yeah, well, thanks, Elaine. Great to have you today to help me with the event. And thank you everybody for coming. I do see the poll results, and thanks for answering that. I see almost half of the audience are interested in germ-free mice and probably already using germ-free mice in your research. That's great. We look forward to hear from you if we can help you in any way. And I'm also seeing people who are saying they have been a frequent attendee to our webinar. Thank you so much for your support. And again, we also look forward to have more interaction with you, science-wise. So I will share my screen to get today's webinar started. Can everybody see my screen? Yes?
Yes.
Great.
Yes.
So yeah, I want to make sure, because once I start projection mode, I won't be able to see people anymore. Okay. So again, just a brief self-introduction. My name is Zhiying Li. I'm the Director of Life Science and Technology at GemPharmatech. Very happy to share with you today about our germ-free platform, including both mouse models and services that we want to bring from our parent company to the US market. A brief disclaimer, and to share with you this, I will have 3 topics for today: an intro on germ-free mice and technology, applications of germ-free mice for life science research, and then finally, I'll share some information about the mouse models and services that we have at GemPharmatech. All right, so very quickly, germ-free, basically there is no microorganisms in the animal, including all types of microorganisms and throughout the life cycle of the mice in our context. And here in this slide, I highlighted throughout the history, or in the historical development of the germ-free technology, there has been major players, scientists, as we all know, as the foundation and the leader for this germ-free technology to become available for today. And as you can tell, Louis Pasteur had a statement thinking that germ-free animal life is not possible. And 2 major group of scientists, including Dr. Gustafsson and also the Lebond Institute, their contribution in the history of germ-free animal development as well as technology, including isolators, has contributed greatly to what we can do today. Again, very briefly, germ-free, is basically a containment or monitoring of microbial organisms in the environment as well as the body of the mice. So there are a few different ways to identify, microbial containment. Here I'm listing one of the very common ones, starting from clean to specific pathogen-free to no-probiotic, as well as the final highest level is the germ-free, meaning no microorganisms. And then facility-wise, the first two could be realized in a barrier facility. The last two will require isolation. A lot of these isolation will use isolators in different types. I believe there has been new technology being developed to support isolation and no-probiotic and germ-free environment. Now, in terms of germ-free mice, we are so used to SPF mice, or the regular mouse, as we talk about mouse models, or the research animal that we use in the lab. So in the next couple of slides, I'll share with you a quick description about what is the characteristic differences between a germ-free mouse versus a typical, SPF or regular mouse. There's a few different ways, as you can tell, GI, cardiovascular, immune, metabolic, reproduction, as well as central nervous system. So in this slide, I'm showing you with the picture and some description, for example, in the cardiovascular system, the most obvious difference is that germ-free mice, they have smaller heart, and this relates to functional differences in these guys. And in the GI tract, one of the most obvious changes is that they have very much bigger cecum. And then in the immune system, their spleen is smaller than the regular mouse, and associated with that, as you can see from my highlights, their lymphoid tissues have some dysfunction. Oh, I see another Paul question come in. So give it a try and let us know your answer. After you answer it, the Paul question, the Paul window will go away. So I'll continue on my part as you answer the question. So in the immune system, there will be some differences. Gut-associated lymphoid structures are different, and there's a couple of structures I highlighted. Yep, and then on the metabolic side, these guys tend to have reduced body fat, and their metabolic rate is reduced. Their water food intake is affected. Their absorption and retention of the macro nutrients are also affected. Reproduction, there are different aspects of reproduction, but one thing I think is quite interesting for us to note is that they have fewer neonates per female. So relatively speaking, they have a reduced reproductivity in the life cycle. So in the CNS, there has been quite some research going on, as well as in other fields. So I highlighted some points, and there are some references I will share with you as we go along with today's presentation. So in our hands, as I'm gonna pause a bit. I think the Paul results is coming back. So interestingly, what in the mouse is roughly 10 times that of a human. We have 70% of audience saying heart rate, 14% saying blood pressure, and then respiratory rate and body temperature. I believe majority of the audience got it right. It is the heart rate. Mouse is about, mouse is about 10 times of that of human. All right, I'm going to close that on my window. All right, so in our hands, we do have germ-free mice in our commercial offering, and we have been characterizing them in our internal R&D pipeline as well. So here, showing as an example to you, our typical B6 mice, we have been comparing the GF versus the SPF. These are data from males. So you can tell morphologically, a lot of organs are affected by this microbial macro environment that the mouse is living in. And at the same time, some biochemical measurements in the blood, in the periphery, as you can tell, are also changed due to the change of GF versus SPF. I want to give a highlight to our NCG animal. This is our foundation line for all the severe immunodeficient mouse strains that we are developing in order to support the IO study. In this perspective, we also developed germ-free NCG. So when NCG is living in germ-free versus the regular SPF environment, there are also fundamental changes that we observed. But showing on this slide is a comparison between our male versus females. And this is the biochemical parameters that we have been collecting data on from these guys. Now, I'm going to move to my second part of today, talking about applications of germ-free animals. So in the application, I'm going to talk about, GF versus SPF, gene function, cause effect observations, which means, the living environment, including what is within the body of the animal, actually affect the physiology of the animals. And I'm going to share some information about, how we are doing live biotherapeutical product exploration and efficacy evaluation in support to our customers. So in here is basically a highlight, microbiome or germ-free animal has been applied and explored in many different research areas. The first one I want to highlight using these two articles is the oncology field. For the interest of today, I'm not going to go into the details, but feel free to take a look into those research and then engage us if you have questions or information that you need from us. So oncology, this is immunology, and then metabolic diseases, as you can imagine, in the GI tract, as well as overall metabolism of the animal, and finally neurobiology, even in Parkinson's disease. I think microbiome or microbiota, how that's interacting with the living organism has been really elucidating a lot of unknown information in our past research. So there's a lot of room for this area going forward. Now I'm going to segue into the last part of today's webinar, which is to give you some information on our germ-free mouse models as well as services. But now I see there's a quick poll coming up on my screen. What is your research interest? So take a look into these areas, and we'll show you the results in a minute or so. All right, so I'm going to talk about mouse models as well as research platforms. So showing on my screen for you, on the mouse model perspective, we do have available germ-free mice we already developed in-house and available through our commercial offering. We have also developed germ-free rederivation technique in-house. Using this technique, that will enable us to generate or develop new GEMS models for you if you already have an existing GEMS model in your hand. And finally, our custom model generation pipeline can be coupled with the germ-free capability to generate completely new germ-free genetically engineered mouse models. On the research platform side, I will introduce some information on the microbiology lab capability. That's really the foundation that enable us to even establish the different health status for the animal. And then using that, we have been generating our own germ-free animals and using them for research activities in experiments. And then finally, I'm going to show you some information about how we use germ-free animal in our immuno-oncology, IO, or metabolic disease research to support the preclinical testing for our customer. I see the poll results showing on my screen. So we have a good spread across all the research area. I'm just going to highlight the top 3: immuno-oncology, pharmacology, and developmental biology. All right, moving on. So, germ-free capability fundamentally is a monitoring and containment on the microorganisms. So in order for that to be a reality, what we need is fundamentally from everything that touches the animal. The foremost is the facility. So we do have a huge SPF facility. I don't have to, take your attention on that. On top of that, we also have developed germ-free or neurobiologic capability in using isolators primarily. We have about 1,300 cage capacity in our current germ-free facility. That's about 2,000 square meter by square area. And on the left side of the screen, I'm showing you some accreditation we have to acquire in order to produce the mice. On the right side is our isolator facility in Rio on the bottom. And then on the top 2 panels is actually, we have automated monitoring system that enable us or our staff member to monitor how the entire facility is working to provide the germ-free environment for the mice. At the same time, to contain the microbials, we have to do sterilization consistently and reliably with everything that the germ-free animal may touch. So I listed the different supplies or materials that we use, and also the testing ways we have been using in our production pipeline for the germ-free. And in this one, we contain it, but fundamentally we need to use testing to make sure it is germ-free. So this is where the microbiology capability is very important. So we do testing primarily in 3 ways: microbiological cultures, and molecular biology, PCR sequencing to see what's the DNA signature of the microorganisms. And we also rely on we also use visual inspection, coupled with microscopes to see what's going on in our colony. And here, I know most of you probably wouldn't be able to read, but it's basically, we have guidelines in parent company that in order to be able to produce and use germ-free animal, there are regulations we have to comply to. So in this one as well, to showing you to comply with the I-class requirements, we have our internal health monitoring program, testing, pathological bacteria, testing viruses and parasites from our animals. And then given the health standards we are testing these animals against, it could be our SPF or our germ-free standard. In this one is a quick summary of the in-stock germ-free mice or strains we offer, from the typical B6 to bulb, ICR, and then our NCG, and one of our next generation NCG-X. This particular strain doesn't require irradiation or chemical ablation in order for stem cell implantation engraftment to take place. So is a very useful line down the line. Last, we have the L10 knockout animal, which can be used in IBD research. So again, we have breeding capability. At the same time, we also have procedure facility to support experimentation and research activity using these germ-free mice. I highlighted on the left side a few of the areas that we apply the mice and our capability for. I'll go into a bit more detail as I go through the presentation. Just a quick, before you start to wonder, we do have our own capability to provide mice to our customers. So we have 3 types. One is the shipper container, which is a steel apparatus. We also have the shipper sleeve, which is built in-house and is based on plastics. And then the last one is our transportation using isolator. This is a very new technique we have been developing in-house, and I believe this is available for our customers in China. I'm seeing another poll coming into my screen. What's a fun fact about germ-free mice? There are 5 different options for people to choose. Yeah, so take a look, and if you can answer the poll, we may get some interesting responses from our audience. So let me continue. So on my side, we have, one of the fundamental techniques in order to enable germ-free offering is the germ-free rederivation capability, which can also be used for our customers to develop their own germ-free mice or germ-free mutant mice. So this one, I'm giving you a few pictures to look at, but fundamentally is under germ-free environment, and we do IVF, and we also do cesarean section to deliver the baby. And now this one, as if you have come to our previous webinars, in particular webinars on KOAP, we have under that flagship program, we have about 16,000 KO or CKO mouse strains already available. So these strains, when they are coupled with our germ-free capability, we can generate a lot of new germ-free gem strains for our customers. So this is like, once they become germ-free, they will be living in our isolators. That's why the cartoon on the right side is trying to show to us. And then all the germs where the microorganisms are blocked by the isolator. And our service team have done this very successfully for a lot of customers in China. All right, the poll result came in. All right, I think everybody heard me or saw the slide. 88% said the fun fact about germ-free mice is that they have a large cecum, which is true. Somebody said they are more likely to put on fat, but the reality is, by statistics, they tend to have a lower body fat percentage. Right. Yep. So here, just picture worth 1,000 words, right? I'm showing you a few enabling equipments in our microbiology lab, which enable us to identify the microbes we have, and then some of the microbes by our internal interest or customers' requirements, we preserve them in order to do transplantation using them, or single microbial colonization on our animals, the GF mice. We can also, qualitatively and quantitatively characterize the bacteria we identify. We do, fecal microbiota extraction, and one of the equipment we use, I highlight here, is called VetTech, that enables quick microbial detection and ID in our pipeline. This is a quick overview of how we use the germ-free in our experiments. So it started with, we have, our operational team enables the germ-free environment for the animal throughout their lifespan, and we use their rederivation, containment to keep their health status. We have transportation methods and system available to move the animals around, and then we experiment on them. So a quick overview. If you have come to our previous webinars, you would know on the service side, we have been offering immunology, immuno-oncology, as well as metabolic disease preclinical services to our customers. So adding on to those capabilities, we are providing services, a germ-free service on top of these 3 research area. And some of the highlights, just to mention again to you, we can do cell culture, which is coupled with cell line engineering capability using CRISPR-Cas9. We have flow cytometry, multiplex in-house. We can also do histology or pathohistology analysis on the disease animals before or after the preclinical testing. All right, coming back to this diagram, in the next few slides, I will give you a little more flavor in the research services that we can provide using our germ-free animals. These range from fecal microbiota transplantation, microbial transplantation or colonization, some of the drug efficacy evaluation we have done in-house, and then disease model construction, as well as additional customizable services that we can provide. So first of all, on the fecal microbiota transplantation aspect, I'm showing you a diagram how we do the transplantation. It started from feces sampling. We do microbiota extraction using the samples. We garage the GF mice and then collect their feces. After that, we identify and characterize what's the microbial that we have collected in these samples. Showing you in the lower part of the slides, on the left side, is a comparison of GF mice on the top versus a GF mouse that has been transplanted with fecal microbiota. So the organs, the first one is a cecum, second one is liver, third is spleen, and last one is kidney. So as you can tell, the biggest organ, the cecum in the GF animal, has been restored, quote unquote, to a normal size after fecal microbiota transplantation. And that data is just graphed in the plot on the right side. Now, coupled with that transplantation, it could be a microbiota, which is a whole community of microorganisms. At the same time, our microbiology capability enable us to isolate a single bacterium, for example, and then we can use that to do the transplantation. And in this way, we really know what we provided to the germ-free animal. And then in this pipeline, this type of research will enable us to learn what's the host versus bacteria effect or the interaction between the two entities. And this will provide information probably on some disease or treatment options. And here is one of the, an example, I would say, in the IO therapeutic efficacy evaluation pipeline is tangled to our preclinical evaluation for IO field. So in this diagram, what I'm showing you is what we can do using a germ-free mouse in order to test a upcoming, for example, a novel or a new IO therapy agent. In this case, we're using the anti-mouse PD-1 as an example. So when we have germ-free mice, what we can do is, in the regular SPF mice versus the GF mice, we give them the same treatment. And then on the right side of the panel, which is tumor volume analysis. So we can evaluate, the different microbiological environment, whether that's going to have an effect on how effective that particular therapy can do. So as a proof of principle, I'm showing you some data that we have generated in-house. On the left side is the germ-free animal, and on the right side is the SPF animal. So they have been engrafted with the same tumor cell line, which is MC38, is a syngeneic tumor cell line. And these mice can both enable the growth of the tumor cell line to become a tumor sub-Q, And when we provide the same treatment using the anti-mouse PD-1, you can see in the colored lines how the tumor growth inhibition has been taking place in these animals. As you can tell, GF mice have a different profile compared to the SPF mice. So the interesting thing would be to figure out what is contributing to the difference between these two groups of mice. Now, using our germ-free mouse strains, as well as the service or research platform, not only we can compare GF versus SPF, but we can also, using the same GF mice, but bring back the microorganisms, either as a single colonization or as a multiple bacterium colonization, and then do the same evaluation. And this respect is, again, to provide more information on how is the microbial community that is contributing or affecting the effectiveness of the anti-tumor therapy. Now, this is a simple summary of what we are thinking in terms of using our germ-free capability to develop new disease models. So we have a few different ways to do it, and we have been looking into, for example, using germ-free or constructing germ-free animals that are has been going through ovarian sectomy and then developing osteoporosis. We have also been looking into metabolic models, which are diet-induced obesity, or they may have colon cancer in this respect, and some other drug-induced disease models in pancreatitis or colitis. And then lastly, we are looking into disease models that have implications for hyperoxia. So this comes to my last slide about today's webinar is a summary. I hope by now I have brought you information in the following few aspects. The first one is, the germ-free rederivation capability or the technique has enabled us to generate GF gems for the customer and for the research need. We also have in-house germ-free strains that we can offer through our commercial offering, B6 BOBs, ICRs, NCGs, and with the NCGX as well, the B6 L10 knockout. We can, on the service side, we can do fecal microbiota transplantation and single or multiple bacterial colonization for customers. We can, combine the germ-free capability with preclinical efficacy testing to offer, evaluation of how the microbiota is affecting the efficacy or safety of the anti-tumor agents. We are also looking into developing disease models that are, germ-free, so that we can look into how the microbial community that has been affecting the disease mechanisms. And lastly, we do have, a lot of ways that we can customize to your research needs on top of our, existing pipeline. So as a last slide to end the webinar, I want to give people a sense what's coming on in the next few months. In March, we're going to talk about one of the models we are developing for Alzheimer's disease. April, immune and checkpoint is a topic. In May, we're going to talk about IOs and bispecifics. And in June, we're going to talk about NASH. So we look forward to have interaction with you, either as a customer or collaborator. We want to say, we really see your research need as a top priority. What we want to do is, we want to support your research and enable the development and the innovation in the research that coming in the near future. So the last slide is our contact, but you can always visit our website, GemPharmatech.us. That's for US customers, and for people in China, you can go to GemPharmatech.com very easily to browse through and get into contact with us. Okay, with this, I'm gonna close the webinar and I'm gonna stop sharing my screen.
Thanks, Zhiying. Thanks for your nice presentation. Let's go to the Q&A session. I did receive several questions from the audience. The first one from our audience is that how long will it take for sending your germ-free mice to our US facility?
I think I think the question is, behind the question is whether there will be a difference between, shipping a regular mouse versus shipping a germ-free mouse. I don't think there's a special or anything that's significantly different between shipping a regular mouse versus a germ-free mouse. So the typical timeline for shipping live mice from China to our US customers, which we have done a lot and very often, honestly, is about once the order is confirmed, it takes about 2 to 3 weeks. That's basically to have the logistics figured out. And we do have our logistics teams that help with this particular aspect. And with the pandemic, I just want to share with everybody, the pandemic has actually making the logistics quite difficult, not just international, but also domestic in the US.
Yeah, thank you, Zhiying. So another question is that, what's the UU frequency of your FMT?
What's the
UU frequency of your FMT?
You mean the how many times do we gavage the animal?
Yeah, the frequency, the cat.
Oh, I see. So we in our research pipeline, typically we do it 3 times throughout the week, and on average every other day, and then we do it for a week.
Okay, thank you, Zhiying. There is another question, maybe the last one. An audience wondering, could you offer a gene-free disease model, such as OB or DB gene-free mouse model? Is this a customized gene-free service you could provide? And also, if I want to perform some phenotyping in your facility, could you take such service?
Yeah, so we do, as most, this customer may already know, we actually offer leptin knockout and leptin receptor knockout. If these are, the 2 particular strains you are looking at, we can, the mice, they are available in our parent company's facility, so we can do germ-free rederivation on these animals. As of now, I would want to say, since they are not as part of our in-stock offering, I have the list in my presentation. Since they're not showing up there, I believe we will have to sort of generate them given this request. And I believe with our phenotyping platform, we can do phenotypic analysis for you. And then it really depends on what this particular customer is looking into. We can figure out what is the parameters that you want to analyze using the phenotyping platform.
Okay.
So, send us an email, sales@GemPharmatech.us. That we can, and also, or if you want to, you can go to the website. We have, contact us, that you can also get into contact with us, and we can help you to figure out, what's can be done, and then the timeline.
Thank you, Zhiying. Thank you all the audience. I think that's all for today's webinar. Thank you again for all the audience who joined us today at this webinar. We're looking forward to seeing you next time. Have a good day.
Yeah, thank you everyone. Nice to see you.
Thank you. Bye.